Cellular transport is the movement of molecules in and out of the cell. It is vital for our cells to survive and for them to carry out important biological functions. When the word transport is brought into discussion, people often think of planes, cars, or trucks that help move our goods from one place to another. But this is biology class, so we can't have pictures of cute trucks and planes. What we do have is where, on a molecular level, molecules constantly move in and out of the cell's plasma membrane. There are two main types of transport, and they are passive transport and active transport. Passive transport is diffusion of a substance across the membrane with no energy investment. It also moves down its concentration gradient, the region along which the density of chemical substances increases or decreases. Active transport, however, requires an input of energy in the form of ATP to help facilitate the molecules across the membrane. It goes against the concentration gradient. Starting with passive transport first, we can break it down into three subsections, which is diffusion, facilitated diffusion, and osmosis. The most important one here to know is diffusion because both facilitated diffusion and osmosis are a specific type of diffusion. We keep talking about diffusion, but what exactly is diffusion? Diffusion is the spontaneous movement of a substance down its concentration gradient. Each molecule is moving randomly, but for diffusion is where the entire population of molecules moves in a particular direction. The molecules move from high concentration to low concentration in order to reach equilibrium. Small, hydrophobic, and nonpolar molecules can easily diffuse due to plasma membrane's lipid bilayer. But how do large, hydrophilic, and polar molecules move across the plasma membrane? The answer is through specific transport proteins that are found all throughout the cell membrane. There are two types of transport proteins, and they are channel proteins and carrier proteins. They allow for what is called facilitated diffusion, where because of transport protein, the movement of water or a solute across the membrane down its concentration gradient is increased. Beginning with a channel protein, the one on the left, it is a hydrophilic channel that is used as a tunnel by certain molecules or ions to move across the membrane at a faster rate. One famous example of a channel protein is an aquaporin, which allows for water molecules to move across the plasma membrane. This process is called osmosis, where water moves from an area of high concentration to a lower concentration. A few other channel proteins are ion channels that allow for ions to move across the membrane. Some can even be gated channels. For the carrier protein is where the one on the right, they are proteins that hold on to their molecules, change its shape in a way that shuttles the molecules across the membrane. It is highly specific and it transports only a certain substance. One example can be the glucose transporter that transports glucose across the membrane. It is so specific that it even rejects fructose, a structural isomer of glucose. One more important function of carrier proteins is that they carry out active transport, which we will explain later on in the video. Back to osmosis, the movement of water across the membrane through aquaporins. This process is vital to an organism's sustainability and balance. Here is a picture of three different types of water balance and its effects on cells. One term we need to know is tonicity, which is the ability of a surrounding solution to cause a cell to gain or lose water. The solutions depicted show three different levels of tonicity. One is an isotonic solution, where the cell is immersed in a solution that is the same and there will be no net movement of water across the membrane, thus making the cell flaccid. Water diffuses by the same rate in both directions in and out. Animal cells do best in these conditions. The second one is hypotonic solution, where the cell is submerged in a solution that has a lower tonicity than the inside of the cell. Hypo meaning less, in which water will then enter the cell faster than it leaves. The cell will swell and lyse, aka burst, as if it were an overfilled water balloon. But with plant cells, it is a different story because they have a cell wall which exerts turgid pressure back on the cell, thus opposing any further water uptake. Lastly is a hypertonic solution where there is more tonicity on the outside of the solution than on the inside. Hyper meaning more, where the cell loses water to the environment, shrivels up, and most likely die. This results in the cell being plasmolyzed, where all the water moves out of the cell and into the environment, making it all shriveled up. Now to the last portion of the transport review, which is active transport. Like mentioned earlier, active transport is where molecules are moved against a concentration gradient, thus making it non-spontaneous and therefore need an input of energy. The way ATP energy powers this is by transferring its terminal phosphate group directly to the transport protein. When this happens, the protein changes shape, which then translocates a solute bound solute bound to the protein across the membrane. An example of this is the sodium-potassium pump. The cytoplasmic Na plus binds to the sodium-potassium pump. The affinity for Na plus is high when the protein has this shape. Then, the Na plus binding stimulates phosphorylation by ATP. Phosphorylation leads to a change in the protein shape, reducing its affinity for Na plus, which is then released outside the plasma membrane. The next shape has a high affinity for K plus, which binds on the extracellular side and triggers release of the phosphate group. Loss of the phosphate group restores the protein's original shape, which has a lower affinity for K plus. K plus is released, affinity for Na plus is high again, and the cycle repeats all over. Now moving on to the idea of membrane potential, which is the voltage across the membrane. A transport protein generates the voltage across the membrane is called an electrogenic pump. For animal cells, the sodium-potassium pump is the major one, but for plants, fungi, or bacteria, they use proton pumps that actively transport H plus from the cytoplasm to the extracellular solution. Which brings me to my next point of co-transport, where a transport protein can couple the downhill diffusion of the solute to the uphill transport of a second substance against its own concentration gradient. With the ATP-powered protein pumps, it drives active transport of amino acids, sugars, and etc. into the cell. With sucrose H plus reaction, the co-transporter couples the return of H plus, thus moving the sucrose into the cell again against the concentration gradient. Now finally we are up to the last type of active transport, which is bulk transport. Bulk transport is where molecules packaged in vesicles are moved in and out of the membrane. This occurs through different processes known as exocytosis and endocytosis. Exocytosis is where the cell secretes certain molecules by the fusion of vesicles with the plasma membrane. The vesicle is budded from the Golgi apparatus in the cell and confused with the lipid bilayer of the membrane. While doing so, the particles then exit the cell. Exo meaning exit. Now with endocytosis, it is where the cell takes in molecules and particulates matter by forming new vesicles from the plasma membrane. There are three types: phagocytosis, aka cellular eating, pinocytosis, aka cellular drinking, and receptor-mediated endocytosis, which is also a pinocytosis. For phagocytosis, the cell engulfs a particle with the pistoponium and packages it with a food vacuole. Then a lysosome comes and fuses it with it, thus digesting the insides. For pinocytosis, the cell gulps droplets of extracellular fluid into tiny vesicles that is formed from the plasma membrane. They are often lined with a fuzzy layer of coat protein known as pits. Lastly is receptor-mediated endocytosis, where cells acquire bulk quantities of substances by proteins with receptor sites and are exposed to the extracellular fluid. Solutes then bind to the sites and bring them into the cell. This is the end of cellular transport, and if you have any questions or concerns, feel free to contact me at lya@bronxscience.edu, and I'll be happy to answer your questions. I'm Angela Lee. Thank you for watching, and I hope you enjoyed the video. Bye!